Welcome to the pod Biomanufacturing and Fermentation Technology. In this episode, we translate microbial science, process engineering, and manufacturing realities into insights that scale.
Imagine you are just hanging out in your kitchen, right? And you've just baked this perfectly complex, multi-layered cake.
Oh, nice. Sounds great so far.
Right. You used like a million complex ingredients. You've got your flour, sugar, specific fats, raising agents, and you bake it to absolute perfection.
Okay, I'm with you.
And here's your end goal isn't to actually eat the sponge cake.
Well, that's just a tragedy, honestly.
It really is, because your end goal is to somehow reach into that baked cake and extract only the pure liquid vanilla extract back out of it.
Oh, wow.
Yeah, while leaving every single crumb of the sponge behind. Just the vanilla.
I mean, that impossible task is actually an incredibly accurate way to picture the headache that industrial chemical engineers face every single day.
Right, because we kind of want to believe our modern medicine is born from absolute pristine clinical precision.
Oh, absolutely. But the reality of how certain critical drugs are manufactured, it completely shatters that gleaming stainless steel illusion.
Okay, let's unpack this. Because to understand why this matters for you listening, we have to look at what we are actually trying to extract.
Right.
Today, we are doing a deep dive into the production of a chemical called mycophenolic acid, or MPA.
Yeah, MPA. And, MPA isn't just some abstract scientific curiosity.
Not at all.
It is a highly valuable, absolutely critical precursor for life-saving immunosuppressive therapies.
Which means, for instance, if someone receives a heart or kidney transplant, their body's natural response is to attack that new organ, right? Like it's a foreign invader.
Exactly. And MPA suppresses that specific immune response.
So it's a big deal.
Oh, a massive deal. There is a very high chance a derivative of this exact compound is what keeps transplant recipients' bodies from rejecting their new organs. The stakes, they literally could not be higher.
But getting a microscopic fungus to actually manufacture this life-saving drug is, ironically, kind of the easiest part of the process.
Yeah, it really is. The real nightmare, the absolute bottleneck of the entire global industry, is figuring out how to get the drug back out of the fungus once it's made.
So to understand why that extraction is so remarkably difficult, we have to look at the biological factory itself, right?
Right. So the industry relies on a specific fungus called Penicillium brevicompactum.
Penicillium brevicompactum. Okay.
Yeah, and MPA is a secondary metabolite for this organism.
Which means, just to clarify, it's not something the fungus makes just to survive its day-to-day life.
Exactly.
Because primary metabolites are the basic building blocks for growth, right? Like eating and breathing.
Precisely that. Secondary metabolites, like our MPA, are complex compounds the fungus only produces under very specific environmental stresses.
Sort of like a panic response.
Yeah, kind of. And industrial data shows that to trigger this production, manufacturers rely on what's called submerged fermentation.
Submerged fermentation, what does that actually look like?
Well, they grow the fungus in massive, building-sized liquid nutrient broths rather than, on solid surfaces.
Oh, wow. Building-sized.
Huge vats. And the goal is to carefully starve or stress the fungus into a highly productive secondary metabolic state.
So you're pumping out MPA without triggering massive biological overgrowth.
Exactly. You want the fungus working and producing the drug, not just getting fat and multiplying out of control.
Right.
And when these submerged systems are highly optimized, modern engineers are finding they can hit multi-gram per liter levels of MPA.
Which sounds like a lot.
It is.
Right.
In the industry, this concentration of the drug in the liquid is called the titer.
Okay, the titer. Wait, so if I'm running a factory, 1,000 units a day is fundamentally better than 500 units, obviously.
Sure, yeah.
Why wouldn't a pharmaceutical plant just crank up the biological production to the absolute maximum to get the highest titer possible?
That is the logical assumption, right? But it leads straight into what process engineers call the titer trap.
The titer trap?
Yeah. So you could push the fungus to produce a massive amount of MPA, but the resulting broth becomes so incredibly thick, messy, and complex that it actually becomes physically impossible to separate the MPA from the fungal waste.
Oh no, so you just ruin the batch.
Basically, you end up with a giant vat full of precious medicine that you literally cannot access.
So it's less like having extra cars sitting on a lot waiting to be sold, and more like trying to harvest tiny beads of water that are super glued inside a massive sponge of steel wool.
That's a great way to put it. Doesn't matter how much water is in there if you can't squeeze it out.
Wow.
A high titer broth that cannot be easily separated is far less profitable than a moderate titer broth that clarifies easily.
Because it just gets stuck.
Right. In industrial bioprocessing, the economics are not measured by fermentation titer alone. The only metric that keeps a manufacturing plant open is recovered kilograms per fermenter year.
Recovered kilograms. So it is entirely about what you can successfully pull out of that vat and purify to a pharmaceutical grade.
Exactly. It's all about the downstream.
Okay, so if the broth is this thick, toxic smoothie that ruins standard filters, how on earth are we actually getting pharmaceutical-grade medicine out of it today?
Well, we are wading deep into the fungal swamp here.
Okay, grab your boots.
Yeah. The downstream processing, meaning everything that happens after the biological fermentation is done, is a total nightmare. And that's because Penicillium brevicompactum is a filamentous fungus.
Filamentous,
Sweet.
meaning it has threads.
Yes. It doesn't grow as neat, floating single cells like a simple baker's yeast, It grows in a complex, tangled mycelial network of microscopic threads.
Oh, so it's like a microscopic ball of yarn that's constantly weaving itself tighter and tighter.
That is exactly it. And that tangled network drastically alters the physical physics of liquid.
Yeah.
Drastically increases the viscosity.
So it gets super thick.
Insanely thick. The broth becomes a non-Newtonian fluid.
Oh, like a slime.
Right. So if you try to force it through an industrial filter, those mycelial threads just compress and create an impermeable layer. You run a massive risk of fouling. Yeah, which essentially means completely blinding your multi-million-dollar filtration equipment. It just clogs, totally solid.
So for you listening, imagine trying to push a thick, pulpy, fiber-filled smoothie through a standard paper coffee filter.
It would tear immediately or just pool up at the top.
Right, but it's infinitely harder than that because you aren't just trying to get the liquid. You are trying to save one specific microscopic vitamin from that smoothie while leaving all the pulp, all the sugar, and all the other vitamins behind.
What's fascinating here is that the physical shape of the fungus directly dictates this viscosity.
Which directly dictates your profit margin.
Exactly. And that smoothie isn't just thick, it's chemically chaotic.
What do you mean by chaotic?
Well, the fungus naturally produces pigments, lipids, and other waste molecules. Plus, you have all the unconsumed proteins and sugars left over from the food you fed the fungus in the first place.
Oh, so it's a total soup, all of which probably love to cling to the MPA and get co-extracted right alongside it.
Oh, they cling to it like crazy.
Honestly, it seems like a miracle we get anything usable out of this at all.
It is a chemical miracle. The established industrial route to actually achieve pharmaceutical-grade purity is essentially a 7-step obstacle course.
7 steps. Just to get it out of the liquid.
Yep, and it relies entirely on a concept called pH-driven extraction logic.
Okay, wait, how does that actually work mechanically? Are we just changing the acidity of the broth?
Essentially, yes. Because mycophenolic acid is, by definition, an acid, engineers can manipulate the pH of the broth to change the molecule's electrical charge.
Oh, interesting. So it's like flipping a switch.
Exactly. If you add a base to raise the pH, the MPA becomes ionized, so it gains an electrical charge. And in that state, it loves water and dissolves right into the aqueous broth.
Okay, so it hides in the water.
Right. But if you drop the pH by adding a strong acid, the MPA loses that charge. Suddenly, it hates water, and it becomes highly soluble in organic chemical solvents.
So we are literally flipping a chemical switch to make the drug jump from water into the solvent.
Yeah.
But wait, you've got this massive vat of thick fungal swamp. How do you even take the first step to get the solids out before you start flipping that switch?
Okay, so step 1 is primary clarification.
Primary clarification.
Right. You use massive industrial centrifuges. I'm talking machines the size of minivans.
Minivans.
Yeah, minivans spinning at thousands of revolutions per minute. And they physically rip the fungal biomass away from the liquid through sheer centrifugal force.
Which has to take massive amounts of electricity.
Oh, an unbelievable amount.
Yeah.
And because of the viscosity we talked about, it still makes it incredibly slow.
Wow. Okay, so what's step 2?
Once you have that slightly clear, but honestly still very dirty liquid, you move to step 2, which is broth conditioning.
That's the pH shift, right?
Exactly. This is where they inject acid to drop the pH, removing the electrical charge from the MPA to prepare it for extraction.
Because step 3 is selective extraction.
You got it. This is where the heavy chemistry comes in. Engineers pump in staggering volumes of organic solvents.
Like what kind of solvents?
Harsh chemicals, like ethyl acetate or butyl acetate.
Okay, so you dump that into the conditioned broth.
Right. And because the MPA's chemical switch has been flipped, it migrates out of the dirty water and dissolves into the chemical solvent.
It jumps ship.
Yep. The two liquids separate naturally, like oil and water. Then you literally siphon off the solvent layer containing your drug.
That seems almost too easy. But the solvent isn't a smart bomb, right? It doesn't only grab the MPA.
Far from it. It also grabs those pigments and lipids we mentioned earlier, pulling all that junk into the solvent layer too.
So your solvent is filthy now.
Very filthy, which necessitates step 4, phase cleanup or back extraction.
Phase cleanup, meaning we have to wash it.
Exactly. You have to wash that dirty solvent layer with a new, differently pH-balanced water solution. The goal is to pull the impurities out while leaving the MPA safely in the solvent.
Wait, we are up to step 4 and we still just have a chemical soup.
We do.
How do we actually get solid medicine out of this?
Well, step 5 is concentration and crystallization.
Okay, crystallization sounds like we're getting close to a pill.
Closer, yeah. You apply intense heat and vacuum pressure to boil off or evaporate the massive volume of solvent.
So you're cooking it down.
Right. And as the solvent disappears, the MPA has nowhere left to dissolve. So it literally crashes out of the liquid and forms solid crystals.
Oh, that's cool. But even after all that washing and boiling, I'm guessing it's still not pure enough for human medicine, is it?
Nowhere near pure enough. The regulatory standards for transplant drugs are incredibly strict. You can't mess around with immune systems.
So there's a step 6?
Step 6 is recrystallization.
You just do it again.
Basically, you take those newly formed crystals, dissolve them entirely in a fresh batch of clean solvent, and crystallize them all over again.
Wow.
It is a purely redundant step, solely for polishing away the final microscopic trace impurities.
That sounds exhausting. And finally, the step that I imagine keeps plant managers awake at night, step 7.
Oh yeah, solvent recovery.
You have to capture all those thousands of gallons of volatile chemical solvents you boiled off in step 5.
And evaporated in step 6.
Right. You have to condense them and clean them so you can use them again for the next batch.
The energy required to continuously distill and recycle those massive volumes of solvents, it is just staggering.
Here's where it gets really interesting, especially if you are looking at the economics of making drugs affordable.
Right, the economics are huge.
The sheer volume of solvent required in step 3 and the utility costs to boil and recycle it in step 7. I mean, those are your absolute biggest money pits.
Without a doubt. Every single unit operation you add to clean up this fungal smoothie creates a new opportunity for yield loss.
Because you lose a little bit of the drug at every single transfer.
Exactly. You leave some in the pipe, some in the filter, it adds up. If we connect this to the bigger picture, it fundamentally shifts how we view pharmaceutical innovation.
How so?
Well, often we assume innovation means inventing a magical new nanomaterial filter or discovering a brand new, highly selective designer solvent.
Like a magic bullet.
Right. But industrial data strongly suggests that downstream innovation is actually about making better upstream choices.
Ah, so you're trying to make the obstacle course easier by showing up in better shape.
Precisely. You have to engineer a cleaner, less viscous broth before it ever hits those massive centrifuges in step 1.
So how do you do that? How do you make the broth less messy?
Well, consider something called immobilized or fibrous bed bioreactors.
Fibrous bed? What does that mean?
Instead of letting the fungus tangle up freely in the liquid and make that huge mess, engineers provide a physical structure like a scaffold for the fungus to grow attached to.
Oh, like growing ivy on a trellis instead of letting it take over the yard.
That's a perfect analogy. The fluid flows past the fungus, extracting the drug, but the biology stays put on the scaffold.
That makes so much sense. This drastically improves product accessibility and lowers viscosity.
Exactly. Or there's another method using advanced process analytics.
Like using sensors to monitor the tank.
Yeah. If you can pinpoint the absolute perfect minute to harvest the tank before the fungal cells start breaking down and spilling even more internal proteins and impurities into the broth.
Oh, you save yourself an entire washing step downstream.
Exactly. You aim to minimize the number of purification stages while maximizing the yield at each step.
Because if your extraction is non-selective because your broth was too dirty, you lose massive amounts of product during those final polishing recrystallizations.
Yep, it's all connected.
You know, to truly put into perspective how uniquely stubborn MPA is, we really should look at a totally different biochemical product.
I agree.
Because it really highlights just how much the physical shape of the organism dictates the cost of the medicine.
Yeah.
Let's talk about ergothioneine, or EGT.
EGT is a fantastic counterexample.
So what is it?
Well, EGT is a highly valuable water-soluble antioxidant, and like MPA, it's also produced at an industrial scale by a microbial fermentation.
But their recovery profiles are like night and day.
Totally different worlds. EGT leaves behind a simple, highly manageable watery broth after you remove the cells.
So it doesn't need that massive multi-step solvent extraction gauntlet.
Not at all. Because it naturally loves water, you don't need all those harsh solvents. You just rely on basic clarification and concentration. It's what the industry calls upstream limited.
Upstream limited, meaning for EGT, the main industrial challenge is simply getting the biology to make more of it.
Exactly. The extraction is so straightforward that if you can use genetic engineering to increase the biological titer of EGT in the vat, you directly and proportionally increase your profit.
You just tell the cells to work harder.
Yep. But MPA is the exact opposite.
Because it's heavily filamentous, it requires thousands of gallons of solvent. It's highly sensitive to its physical shape.
And it is completely downstream limited. For MPA, the separation train is vastly more complex than the biology.
So for EGT, our engineers are acting like personal trainers.
Yeah, pushing them to do more reps.
Right. They are just trying to get the cells to work harder, lift more weight, and pump out more product.
But for MPA, the cells are already producing plenty of the drug. The engineers have to act like a hazmat cleanup crew dealing with an incredibly sticky, toxic spill.
That is a wild mental image.
What? It's true. And this raises an important question regarding what happens when you try to scale these processes up from a small laboratory beaker to a global manufacturing facility.
Because stirring a 1-liter glass flask on a lab bench is fundamentally different from mixing a 100,000-liter steel silo.
It is entirely different. When you move to a massive industrial scale, you face physical realities specific to filamentous fungi.
Like what?
Well, to mix a tank the size of a building, you have massive metal impellers, basically giant blades spinning at the bottom.
Right.
And to force that incredibly thick, viscous MPA fungal swamp to circulate, those impellers had to spin with immense force.
Which creates a lot of stress, right?
Huge amounts of mechanical shear stress near the blades.
While the broth at the very top of the tank is probably barely moving at all.
Exactly the problem. This creates a highly heterogeneous environment.
Meaning it's not mixed evenly.
Right. The fungus near the blades is literally being physically battered to pieces, while the fungus at the top is suffocating from poor oxygen transfer.
Oh, wow. So the environment inside the tank is totally chaotic.
Yes, and this mechanical stress literally alters the DNA expression and the physical morphology of the fungus while it's growing.
Wait, really? The physical spinning changes its biology?
Yes. The strands might clump tighter or break apart in response to the stress, which then further increases the viscosity of the broth.
That is wild. The mechanical stress of the mixer changes the biological shape of the organism, which then breaks the chemical extraction process down the line because the broth becomes too thick to filter.
It's crazy, right? The biology and the physics are inextricably linked.
They can't be separated.
Nope. Poor mixing leads to worse morphology, which leads to a thicker broth, and a thicker broth makes clarification harder, which requires more solvent, which ultimately decreases your overall yield.
What is cascading failure?
Exactly. Scale up for MPA has to be treated as a coupled upstream-downstream problem. You cannot look at them in isolation.
So what does this all mean? We started this deep dive talking about the pristine illusion of pharmaceutical manufacturing, and we've ended up in a highly viscous, solvent-heavy, 7-step fungal swamp.
Where the speed of the mixing blade literally dictates the cost of a transplant drug.
It means that the future of manufacturing these vital medicines, the key to making them more robust and globally affordable, relies entirely on a concept called integrated process design.
Yes. You cannot have the biologists in one room tweaking the genetics and the chemical engineers in another room trying to figure out how to filter it.
Right. You cannot tweak the biology without thinking deeply about the physical reality of the cleanup.
It's all one continuous system. Success requires 4 absolute pillars.
Okay, lay them out for us.
First, you need a high enough biological titer. Second, you need a short cycle time to maximize your facility use.
Got to keep things moving.
Right. Third, you need highly efficient broth cleanup. And fourth, you need low purification costs.
High titer, short cycle, efficient cleanup, low cost.
Yes. If any single one of those 4 pillars is ignored, the economic viability of the entire manufacturing plant deteriorates.
It's a house of cards.
It really is.
And for anyone interested in biotechnology or just how our world actually works, the lesson here is really about critical thinking.
Absolutely.
We get so caught up in the glamorous, headline-grabbing world of programming DNA, like computer code, we forget that the output of that code is physical. It's sticky, it's messy, and sometimes it's almost impossible to filter.
The downstream cleanup is not a secondary issue to be figured out later. It is the absolute core of industrialization.
Because learning to respect the gritty, physical reality of separation science is what actually brings these miraculous drugs to the patients who need them.
Beautifully said. It's the gritty reality that saves lives.
Which leaves us with a final thought for you to mull over today. We've seen how the sheer physical shape of a microscopic fungus and the thickness of its broth are the true bottlenecks holding back cheaper, more efficient manufacturing of these critical drugs.
Yeah, the physics gets in the way.
Right. So if the extraction is the real nightmare, could the next massive paradigm-shifting breakthrough in global pharmaceuticals have absolutely nothing to do with biology or gene editing or DNA?
Oh, that's an interesting thought.
Could the key to the next generation of medicine actually lie entirely in material science, in finding a purely mechanical, physical way to safely untangle a microscopic web?
It's a fascinating prospect, honestly. The future of medicine might just belong to the engineers who can untangle the swamp, rather than the biologists who grew it.
Thanks for joining us for this deep dive. We'll be back soon to explore more of the hidden, unseen systems that shape our world. Take care.
You've been listening to Biomanufacturing and Fermentation Technology with Prasad Urnala. Thank you for joining me. Until next time, keep engineering biology into reliable manufacturing.